Scro is our speaker today. She is an expert in molecular tools and marine pathogens and specializes in marine invertebrates. She's currently at Roger Williams University in Rhode Island and wears a lot of hats. She's a molecular researcher, a lab manager, and also a adjunct faculty member who teaches a lot of courses. She actually did her undergrad at Roger Williams University and worked there before coming here to us at UF, where she got a master's in fisheries and aquatic sciences. And that's the work that you're going to hear about today that happened in the Gulf of Mexico. So thank you, Abby, for being here and giving us this last closing out our spring webinar series. And thanks everyone else for being here. And I will turn it over to Abby.
Awesome. Thank you so much, Laura, and thank you, Savannah. Thank you guys for having me. Let me get my screen shared for everyone, and we will get started. Awesome. Well, thank you all for joining. I'm really excited to talk about this work, mostly because we did just get it published within the last year. So this was part of my master's thesis at UF with Don Berenger and a postdoc at the time, Jamie Bojko. And we were really curious about potential pathogens of the bay scallop, with the declines of bay scallops kind of being the main driver, and curiosity kind of got the better of me. I had been working with marine pathogens up here in the Northeast for a while, and I was wondering if there was any maybe potential pathogens for the Florida bay scallop. So let me make sure everything advances. Great. So Florida used to be home to a commercial fishery, and there were actually commercial fisheries of the bay scallop up and down the East Coast. And you can see here on the image on the left-hand side, all of the dark gray areas represent populations of bay scallops. And what's interesting is that there's actually a number of subspecies that make up this population of Atlantic bay scallops. There is Argopecten irradians concentricus, and that is the bay scallop that I focused on. So that's what you guys mostly find during your recreational scalloping, and that's on the Florida Gulf Coast. There is also Argopecten irradians taylori, which kind of diverged from concentricus, and those are in Florida Bay. And there's another subspecies that are on the Texas-Mexico Gulf Coast. And I just kind of like to talk about that because I find it very interesting that the bay scallop has kind of separated and is now they're like genetically unique populations, so they're not as connected as they once were. The commercial fishery actually had a number of landings each year with like probably an average of 100,000, 115,000 kilograms of meat. However, that commercial fishery closed in 1994 because of a number of different factors that caused the bay scallop population to decline. So now we just have the recreational fishery, which is here in Florida from Gulf County to Pasco County, in these counties that are outlined in gray. So on the right, or on the left, excuse me, you see our commercial landings from that commercial fishery, and you can really see how there was just a steep decline all the way up to the closure in 1994. And this is for a number of reasons, overfishing being one of them. We went out there, we collected as many as possible. And it's kind of before we really started understanding some of the population dynamics. And overfishing could still be a threat today to the bay scallop because of the timing of the recreational fishery. And we go out there and it's a fun summertime activity. It's great. I mean, I recommend everyone try it out, but bay scallops have a really short life cycle. So they're only around for about 2 years, and they spawn or reproduce every year in the fall or winter. So we're going out there right before the adults are ready to spawn and kind of removing those adults from the population and reducing the number that could help populate the next year class. Bay scallops are hermaphroditic, but it's not likely that they can self-fertilize, so they need a number of individuals to be able to get to fertilization. So that kind of leads into some poor recruitment. That is another threat to the bay scallop. And finally, another major threat to the bay scallop has to do with the seagrass population. So seagrass is their preferred habitat, especially in Florida, and it's kind of patchy now up and down the East Coast as well as in the Gulf. And that's for a number of different reasons. We've seen seagrass declines because of red tide, urbanization, so pollution, other anthropogenic effects like crop scars, and disease. There is like a slime mold disease that actually affects the seagrass beds in Florida. So for my work, my objectives were kind of to characterize any and all potential pathogens that we found in the bay scallop and create these pathogen profiles. So like basically look at the symbiotic or potentially pathogenic communities within the scallop tissues. And then we wanted to look at how the density of the bay scallops plays a role in that pathobiome. And so you might be wondering, what is a pathobiome? It's a relatively new term, but it is basically the set of host-associated organisms that encompass prokaryotes, which are bacteria, eukaryotic organisms like us, or viruses. And so eukaryotes are like invertebrates, vertebrates, a wide range of different smaller organisms as well, microscopic organisms. And so these organisms together might influence or drive different disease processes in their host, which is the bay scallop. It's kind of a bigger term. It's like a new One Health term. And so it takes in a number of other things into consideration. So stuff like climate change or pollution or, invasive species, those are all going to have an effect and play a role in whether the host becomes susceptible to some of these symbionts or pathogens. So I was really fortunate to be able to work with FWC at the time. There was a scallop team that was based out of St. Pete, and they do all of the preseason and postseason scallop surveys. And they're probably wrapping up right now since the fishery opens in just a few days in Taylor-Dixie County. So the way they do their surveys, they are 100-meter belt transect surveys. And I conducted those in 3 different locations along the Gulf Coast. And those were St. Joseph Bay in Gulf County, the outflow of the Steinhatchee River, which is in between Taylor and Dixie County, and then down off the coast of Hernando County. And so during each transect, we counted and collected all scallops within 1 meter of the transect. We also noted benthic habitat coverage, the different species of seagrass, and took water quality parameters. I'm not going to really get into that too much today. We went to anywhere from 21 to 30 different stations per each of those locations. And then I took a combined 50 scallops from each location. I actually didn't get 50 from Hernando County. We only got 16, but we had aimed for 50 from all 3. And I would bring those back to a lab. Most of the time it was a mobile lab. I kind of set up where I could and created like a sterile environment. And I was able to take tissues and weight metrics as well as, size metrics as well as weight. And these surveys now were conducted 5 years ago. So I guess it might not be what is found in the bay scallop exactly today. However, I'm pretty certain the symbionts I'm going to show you guys are likely common still today. So for the tissue sampling, we wanted to focus on almost all of the tissues of the bay scallop. So everyone likes to eat the adductor muscle, and that is right here. But the animal itself is much larger. So you have these 2 dashed lines, and these kind of represent the area of the tissues that I took. And if I cut on either side of these lines, I created this stake. And that was super useful because in that stake, I had all of those tissue types: mantle, gill, digestive tissues, gonad, and the adductor muscle. And I could take those tissues and I would fix them in a formaldehyde-based fixative, which helps make all the proteins rigid. And then I could use histology for my diagnostic tool. Or I took those tissues and stored them in ethanol and was able to use them for a molecular diagnostic. So molecular diagnostics are your PCR or your next generation sequencing. PCR, you're looking for specific regions of DNA from a pathogen if it's present in those tissues, kind of like we all experienced COVID, so it's a really good reference point. But, you took your swabs of your nose and you sent those out and you got like a qPCR positive or negative result back from the doctors or, the pharmacy. Same idea here, but instead of looking for a virus in a human nose, I'm just looking for pathogens in scallop tissues. For the histology is an older tool, but an extremely useful tool. After those tissues are fixed in that from formalin-based fixative, you can then embed them in like a wax, and that wax allows you to cut really thin sections of the tissue. You will mount them on microscope slides, then stain them, and then view those tissues under a microscope. And so you can see healthy tissues or abnormal tissues. You can also get an idea for what the host's response is, as well as any foreign invaders. So having those two tools together is a bit more comprehensive. To answer my density question, FWC had a project that was ongoing in St. Joseph Bay where they were collecting scallops both during pre and postseason surveys and then holding them in these cages for a period of time, at least through the spawning season. I was able to collect scallops from those cages in February 2020. And so for some of my samples, they were stored in those cages from anywhere from 6 to 10 months. These cages were a half a meter cubed, and they were raised off the bed of the seafloor, and they're in a no entry zone, so there's no boat traffic or anything, but they were stocked with anywhere from 125 to 750 scallops per that half a meter cubed. And that is an artificially high density of scallops. I'll be able to show you guys what the natural densities are from the surveys, but that is 125 scallops in a half a meter cubed is still very, very high dense. And so the reason being is because pathogens tend to thrive in dense environments, like when their hosts are closely packed, which is why, again, if you got COVID, you were told to isolate so that you wouldn't pass on that virus to someone else. Same idea in a marine or aquatic environment, probably even likely more connected when you think about marine environments versus terrestrial environments. So I first am going to introduce you guys to all of the symbionts or potential pathogens that I observed, and basically focusing on like the larger groups. I don't have many that I was able to identify down to species. I got a couple down to genus. And this is an image, a histological image that you can see, and you'll see a lot of these in just a second. So basically introduce you to the players and then talk about their prevalences in the fished range of the Bay Scallop in Florida. So just to orient you to this figure, we have the scallop and then the different tissue types in which I found some of these organisms. And I've labeled them counterclockwise, which is a little counterintuitive, but basically you have A through I. And these are all histological images. So these are all of the organisms I was able to identify histologically, but I also found some using the PCR and molecular tools, and I'll talk about that. Species are difficult to distinguish from one another, and you can see right off the bat that there are a number of different species that actually come from the gills. And so we think about bay scallops, we know that they're filter feeders. So gills are a first line of defense. And so that's often where you're going to find a lot of these symbiotic and or pathogenic organisms. The gills, and I would say also the gut, if they pass through the filter process, they would go further down the digestive tract and enter into the gut system. So first things first, I was able to identify bacterial plaques, and they stain this really dark, dark purple, and that creates a nice contrast compared to the surrounding tissues. This is connective tissue that's right along the mantle. And so we found bacterial plaques in all of the tissue types. We did not see any other major pathology, meaning that the scallop did not have any reaction to these bacteria. There was no increased inflammation or the creation of like granulomas or cysts. So it is likely that the bacteria that we found were symbiotic. Another symbiotic organism was found in the gills. So this is an apicomplexon organisms. They kind of look like little pills. They have been found previously in bay scallops, and they are known as nematopsis. And they kind of get into the filaments of the gills, and they are normally symbiotic, but they can be pathogenic at higher densities. So you can imagine the more you have in those gill tissues, the more they're going to disrupt the process of the gills and or cause ruptures and or tears in these tissues. Something else that was found in the gills that's kind of related to the apicomplexons are ciliates, and you'll see that later on in my talk. Ciliates were also symbiotic. There are gut and gill ciliates. They're very, very hard to distinguish down to lower taxonomic levels because they morphometrically look very similar. So you would need to do some molecular diagnostics or sequencing to really identify them down to species. So next thing are the helminths or the worms. These are pretty cool to me. I mean, most people don't like to know that there are worms and what they're eating. And there is actually a worm that affects sea scallops and infects their adductor muscle. Um, and I mean, that one might make me feel a little bit less likely to eat a sea scallop, but they're not found in bay scallops. So that's a good thing. We didn't actually find any symbionts in our adductor muscles of the Florida bay scallop. So for those of you that like to eat the bay scallop, there's nothing in there, no creepy crawlers or anything like that. But anyway, for the worms that were found in the bay scallop, we have larval cestodes, commonly found in the digestive gland. So the digestive gland is kind of like your pancreas and gallbladder and everything combined. And you can see here, let me see if my pointer works. Yeah. So this round bit is a cross section through the worm. And you can see how there's a couple different points. And so that doesn't necessarily mean there's a couple different worms. It could just be one worm that's like kind of coiled in there. But the cestodes did insist and cause a host immune response. And that's what you see all this light pink surrounding it. That is a granuloma. So the host is reacting to those worms in their digestive gland. So it is causing some harm here. Next up, we can go to this trematode. So trematodes were often found in the mantle and connective tissues. They also form cysts, but these cysts are melanized. So they have like a dark staining outer ring to the tissues. Trematodes, again, there could be a number of them in here, and there likely is based on how many cross sections we see, but it's very hard to distinguish or identify anything further than that. Another trematode was found in the gills, and I'm going to talk in detail about this one. This is pretty cool, but it pretty much infiltrated some of the gills, and this was found in St. Joseph Bay scallops only. And then finally, we have the nematodes, and they're found in gonadal tissues. Here, this is the female gonad, so you can see developing eggs. And this here is your nematode. And then we also had nematodes more commonly in the gill tissues. Now, the gill tissues didn't show any host immune response, but the nematodes in the gonads could be pathogenic because some of these larger helminths, they have been shown that to castrate their bivalve hosts. And so even though we didn't observe any tissue damage, there is the potential for that. Finally, our crustacean symbionts. These are probably a little bit more, are also harmful. So basically the larger eukaryotic organisms tended to be less likely symbiotic and more likely pathogenic or parasitic. Here we have an isopod, and you can see the carapace slats and a brood pouch, and this is in the gut. So like in an intestine lumen or, open space there. And then we also have likely copepods in the gills. So these could be commensal or parasitic. They do cause localized tissue damage because there's usually something they're using to hook onto the tissue. So you're going to break some of the tissue there. You can just imagine if you're a scallop and you have these copepods all in and around your gills, they're gonna cause some damage because they're just large. And so they're larger than the spaces of the gills that are present. This is just going a little bit more detail. This scallop the scallop here had the trematode, and this digenean trematode you can see with the naked eye. This was the only symbiont pathogen that I could see with the naked eye. This is definitely a pathogen, I would say. And it infiltrated the gill tissues. So this is what you see macroscopically, and this is what I see under the microscope. And what it's actually doing is it's getting inside the vessels of the gills, expanding those vessels, and then reproducing within those vessels. And then once they get large, those vessels will pop and release these young trematodes, which are generally a cercaria, and that enters into the water column and or the next host. So a lot of these worms have complex life cycles with multiple hosts, and so that becomes a factor maybe for management. So let's look at these as like profiles now, like, how much of each of these pathogens was found in each location. So just a reminder of my 3 locations, and then give you some idea of the natural scallop densities from those surveys. So Hernando County had very few scallops. There was about 2 scallops per 200 meters squared. And our most dense populations were St. Joseph Bay with 36 scallops per 200 meters squared. I calculated an analysis of similarity. So basically, I'm going to talk about the communities of the symbionts in those scallops. And so technically from the scallops from all 3 locations, we saw a significant difference in the symbiont communities using this analysis of similarity. However, this R statistic, if the R statistic is closer to 1, that means the groups, so like Hernando County and St. Joseph Bay, would be more dissimilar. All right, so you want to see something that's closer to 1 to say that these are really unique populations. But mine's closer to 0, suggesting that I have a more even distribution of the symbiont communities in the Bay Scallop. If we look pairwise, we find that most of this analysis of similarity was driven by the Steinhatchee versus St. Joseph Bay comparison. But again, not a high R statistic, so it's not likely that the symbiont profiles varied very much. This is an NMDS plot. And basically, it takes multivariate data down to like a 2-dimensional or ordinal view. And so this also kind of tells us the same as what the R statistic, just a little bit easier to visualize. So the closer 2 dots are to each other, the more similar they are. And the further away, the more dissimilar. So we see that there's probably some variation within the symbiont profiles by location, but it's going to be very hard to kind of tease out. And so here on the left, we just have a bar graph with percent prevalence on the y-axis and the different pathogens that I focused on, or symbionts, along the x-axis. And so I also took a look at a measurement of diversity. And so I used the Shannon-Weiner Diversity Index, and that usually ranges from 0 to 5, probably typically see anywhere from 1.5 to 3.5. But we see a very similar diversity index, but Hernando County is the least diverse. And that kind of makes sense because we didn't see all of the pathogens in Hernando County. And when we focus on those ones that are most likely pathogenic, we see that, and these are our crustaceans and our worms, basically. But we see that St. Joseph Bay has a higher prevalence of the nematodes as well as the trematodes. And these 2 categories on the right side, those are what I tested for using the PCR. So Mycoplasma is a bacteria, and then I was able to test for other trematodes using a molecular diagnostic. The only different trend I really see is that Steinhatchee had higher levels of your parasitic crustaceans than the other 2 locations. The only thing that might be interesting to remember about Hernando County is that we actually had 100% prevalence for the nematopsis. in the gills. And so those were those apicomplexan organisms that could be potentially pathogenic at higher concentrations or higher numbers in the gills. And we had 100% in Hernando County. So that might be the one thing to look out in that area. Looking at the density, so I pooled all of my wild caught scallops together, and so that's here in the blue, and then all of those cage scallops, and that cage work again was done in St. Joseph Bay. I had to bring this into a 3D plot just because of the NMDS needs a low stress, and mine was too high when I plotted it in 2D. So this is just an easier representation. And what we found was that symbiont profiles were significantly different in wild versus cage scallops. And we now see a higher R statistic. And you can kind of now see how these profiles are like grouping, right? We see that the cage scallops are all similar to each other and the wild caught scallops have similar symbiont profiles to each other. And I did pairwise analysis of similarities as well. In each of the wild caught locations was significantly different from the cage scallops. Now, what we look at here, our diversity Shannon-Weiner index is very, very similar in wild and cage scallops. But now we've got that introduction of the Didymyzoidae trematode. And that trematode, I believe, has actually been identified down to species. We sent some samples over to UNC Wilmington. And it's really interesting because that trematode was genetically similar, near identical to the trematode they found in North Carolina. So they have a problem with these trematodes in North Carolina Bay scallops, which are even a different subspecies, and they were genetically identical to the ones that found in like this near enclosed bay in, the Gulf Coast of Florida. So that means something's connecting those populations. Um, Basically, what we found is that a lot of the larger eukaryotic organisms were in higher prevalences in the cage scallops. So those high density areas had more pathogens, at least when it comes to the pathogenic symbionts. The opposite was kind of true for some of those symbiotic or commensal organisms. We have your gill ciliates, your gut ciliates, and bacteria were all higher in the wild scallops. I'm sorry, not bacteria, the nematopsis. So we're seeing differences in pathogen profiles by location and with density. So it's something to kind of think about. Basically, we found that bacteria, the ciliates, and the nematopsis really had low to no host impact. And so they're most likely symbiotic. So if you see these, if you do any work similar to this survey, if you see these, they're probably not causing any damage. The larger eukaryotic pathogens are likely causing damage. So that's going to be those crustaceans, the cestodes, trematodes, and your nematodes. We found 11 pathogens roughly during this study. We actually found a couple viruses too. They didn't seem to cause any harm. That was through like our next generation sequencing. And so that was interesting. But what does this really tell us? Well, we know that the scallops from Hernando County had lower pathogen diversity. There was really only 5 out of the 10 pathogens that were found in Hernando County. So we're kind of wondering why that might be. And you can start to look at maybe water flow dynamics. So this comes from a study done in 2014, and anything that has an arrow has a net surface flow. And the length of the arrow is a stronger flow versus a weaker flow if it's longer or shorter. The gray circles means that there's hardly any flow, so more like stagnant water. And the boxes that you see are the areas of study for this work. It wasn't part of my work. But it's kind of interesting, if you look at Hernando County, you see there's more flow than what you find maybe in the outputs of the Steinhatchee River. Those were relatively stagnant areas. St. Joseph Bay is also relatively stagnant, even though there's not a dark circle here. And so what this means is that maybe not a lot of scallops can reside in some of these high flow areas. And so when you have lower density, you're going to have lower levels or lower prevalences of some of these potentially pathogenic organisms. Also with lower densities, especially when it comes to the worms, they most often break free of the bivalve host and like basically rip up the tissues. So they enter in the water column. When they're in that water column, if you have high flow, they're just going to get washed away, probably to an area where they don't have another host, and so they're likely going to die, which might be why we didn't see any nematodes or platyhelminthes in the Hernando County scallops. Now, I just want to remind everyone that I only had 16 scallops from that location, where I had 50 from the other 2 locations, so that could have played a factor too. And the last thing kind of is brought on by that Didymyozoidae trematode, and the density in the cages. So we found that trematode in the high density areas of St. Joseph Bay in those caged scallops. It's an almost closed system. You have high densities, so each scallop probably has a lot less water flow that they're coming, or water they're coming in contact with. With these larger eukaryotic organisms, scallops or mollusks in general are likely an intermediate host. So here on the left, we have a trematode life cycle. The trematode basically enters the water column through like fecal deposits, survives for a period of time in the water column. You can imagine our bay scallop being in place of this snail. They're going to filter feed and take out any of those pathogens, filter them. They're going to enter into the gills, which is what we saw in our scallops. They're going to continue to develop, create these cercaria, which are then either released into the water column, or in some cases, like that nematode in the sea scallops that I mentioned, that nematode's life cycle requires a sea turtle, because sea turtles will eat the sea scallop and ingest then that next life cycle, not life stage. So something like a pelagic fish, or a migratory bird species, is likely connecting our bay scallop populations to those North Carolina bay scallop populations. So there are implications. And I do believe the density project, or the cage project, helped to populate the next year class of bay scallops in St. Joseph Bay. They had the highest density of scallops of my 3 locations, and FWC had been doing that project for a number of years. So that's great, but when you are keeping organisms in such a high density, you are risking the spread or higher intensities of some of these pathogens. So yeah, so I'd just like to thank my funding source, and a huge shout out to the team I worked with at FWC, Jen Graneman, and the rest of them. I don't know that the team is still active. I think there's still a group doing some surveys, just not the team I worked with personally. And I will open the floor to any questions. Thank you.
Thank you so much, Abby. That was great. Does anyone have any questions? If you do, you're welcome to unmute yourself and ask your question directly. If you'd prefer, you may also type it into the chat. I can start.
Sure.
I was thinking about the scallop sitters type programs while you were talking about this whole time, and you got to some of the conclusions at the end. But I was wondering a little bit about, like, are all of these organisms acquired environmentally, or can they be passed on through larvae to their young as well?
That's a great question. Most of them are acquired environmentally, and just because, bivalves as filter feeders, they can really take in a lot. They can sort a lot, which is great,
Mhm.
but that's why we see so much in the gills. Even the organisms that tend to infect the gonads, they're larger organisms, so they're not really going to get passed on through an egg or something like that.
Okay, so these big cages, they might increase potentially pathogenic organisms um, close by, but or through birds, but not through individuals.
Yeah. Yeah, mostly through the close by. So like the fact that we had higher numbers of trematodes, cestodes, and nematodes in St. Joseph Bay really has to do with the fact that it is kind of a closed system. They'll kind of reside just in there, because there's higher densities, they have more hosts. And so, if they enter the water column, they'll probably find another host very, very soon after and not have to search around like they might in Hernando County, let's say. I'm in such a fluid environment, everything's so connected. I mean, some of these organisms can survive for, 10 days in the water column.
Cool. Savannah has her hand raised. That's a question.
Oh, you can take the one that's in the Q&A box first.
Sorry, I missed that. I have the chat open, but not the Q&A. Do you plan to follow up with water quality symbiont studies? What type of water quality parameters would you link to pathogens or use as proxy?
Yeah, so I'm probably not going to do further evaluations on the symbionts. The study was kind of designed to see, like, if anyone ends up having, see a mortality event or something like that, there's documents of, what could be a potential potential pathogen that you could look for if it's not a water quality issue, let's say. Some of the water quality parameters we looked into are your DO, your temperature, your salinity, the depths, because there was some variation, even though they're mostly shallow water organisms. You can do, so I work with oysters and clams up here in the Northeast mainly. And one of the major issues for oysters are bacteria that are also human pathogens. So when humans eat raw or undercooked shellfish, they're going to get Vibrio or E. coli, in which case I would use a coliform test for the water and have done in the past. I don't know of a lot of cases in which there has been similar bacteria in bay scallops. So I'm not sure it would work for that. But part of my job here now is to develop new diagnostic tools. And I am looking at a new water filtration system. And actually, I'm working with a company that's has developed a robot to help us actually sample water and be able to bring it back to the lab, extract the DNA. It's like an eDNA kind of project, and be able to see, if you have any of those potential pathogens in the water, and if for your shellfish, you can remove them from the water column. This mostly helps out like the aquaculture industry and farmers. But if you can remove them from the water column while, let's say, like a harmful algal bloom passes, like we could be able to sample and test specifically for that harmful algal bloom, surveying large volumes of water using this robot, and then be able to, give management and aquaculturists that heads up basically before the bloom happens. That would be a goal. So there are different ways. It kind of all depends on what might be a problem in your area. And then I see another.
Yep. The next question in the Q&A are, what are the implications of your work with the cages increasing pathogenic organisms to aquaculture, both presence of pathogens within aquacultured organisms themselves, as well as organisms living near the aquaculture sites? And how much do various shellfish species, symbionts slash pathogens overlap? Scallops spread pathogens to clams slash oysters.
Great question. Some of these larger eukaryotic organisms might spread from clams to oysters to scallops, but a lot of these pathogens or potential pathogens often have host specific, are very host specific. So some of the things that I find in a clam might not be found at all in scallops or oysters. While we think of bivalves as being all very similar, a lot of these species are going to be very host specific. Nematopsis can be found in a couple of different bivalve species, as an example. Some of these trematodes might be able to infect multiple species. In our case, the Didymyzode trematode was able to infect different subspecies of the bay scallops. So there is that potential. In the areas that I surveyed, I'm not sure what type of aquaculture occurs. what type of aquaculture practices. You know, in Cedar Key, I know that there was some oyster and clam aquaculture, but other areas, not 100% sure. And these pathogens or potential pathogens could affect anything that's aquacultured, right? So anything, if you have a wild population that has an outbreak of disease, you know, and you see a die off, it would be important for someone to document that and get an understanding of what's going on there if there's like an aquaculture lease right nearby. Where I am in Rhode Island, we have salt ponds. So Florida has a lot of, you know, fresh water areas, the springs and stuff. So like very karst topography. In Rhode Island, we have something similar, but they're all saltwater ponds. And it's an area where a lot of people do oyster aquaculture and hard clam aquaculture. And they kind of create this like closed off, unique environment that have wild and aquaculture leases. So if something happens in one of those ponds, it's going to affect everybody in the pond because they're, you know, not really large. There's not a lot of flushing of the water in and out. And so, yeah, so there could be broader implications for some of these cages. And I mean, the cages from that FWC used, I think did play a role in the fact that we saw higher numbers of pathogens in the wild scallops as well.
Savannah,
I
your
actually
hand
have
raised
two
one
questions.
time.
I'll start with my scallop related question.
Okay.
So it's kind of similar, I think, to the first question you had, but we know that scallops are really easily stressed by changes in salinity and also like, especially the ones you try to keep in captivity, temperature spikes can be really, really harmful to them. And so I was sort of wondering if any of these pathogens tend to be correlated with either higher temperatures or higher or lower salinities, and like if some of these pathogens might be adding insult to injury for some of those already stressful conditions for scallops.
Yes, they definitely can be. Off the top of my head, so I did take all of this information, and my thesis was split up into a couple chapters. So this was all kind of like the first bit, and we're gonna get more into the ecology side of things a little later. But I salinity and like, you know, poor water quality are also going to affect your bacteria as well as some of these eukaryotic organisms. So I think if you're having a water quality issue and you also have all of your gills have that trematode in them, if you're a bay scallop, it's going to, you know, have compounding effects and probably be difficult. I will say a lot of pathogens are hardier, right? So they kind of can live in probably more extreme environments or at least survive for a period of time. So even if their scallop dies in a like a low salinity environment or, you know, a high temperature environment, that pathogen would be released into the water column and might likely survive where the scallop doesn't. So, and then it can go infect a new host. Did that answer your question?
Yeah, no, that's interesting. I'm sure there's like feedbacks and complexities too of like, was it existing and then it just took hold more because the scallop was stressed versus did it get infected because the water quality was bad? So I'm sure a lot of that was not known at the moment.
Yeah, we didn't. That's a tough one because then you'd have to be looking at the water quality prior to the collections as well, right? Like
Yeah.
long-term studies.
And then my second question is related to kind of a throwaway comment you made right at the beginning about a slime mold disease that affects the
Oh yeah.
seagrass. And of course, we are a very seagrass heavy group. So I kind of just want to know more about that seagrass slime mold and kind of the same question of, are there certain conditions that make that more likely to become a problem for the seagrass?
So the slime mold is like a labyrinthola, I believe it is. So some people like to call it labby. Um, the conditions in which it thrives, I'm not 100% sure, though I do know there's a group that is studying that that disease as well as like an eelgrass wasting disease and looking at the cohabitation of a bivalve, a clam, I think it's a clam, and those seagrasses to kind of see if having bivalves present in seagrasses help to filter out that slime mold and or whatever other pathogens might affect the seagrass beds. And so that is possibility, though, you know, it could kind of go both ways. I believe I saw a presentation about that where it both harmed and helped the seagrass a little bit. So it's not necessarily understood fully. And the slime molds are definitely interesting because they kind of, you know, coat the seagrass and, you know, they'll brown and then it causes them to, they can't photosynthesize anymore and they'll die off. And then, I mean, for our case, that would definitely hurt the scallop then too.
Yeah.
But yeah, so a labyrinthola, eelgrass, the wasting seagrass or eelgrass wasting disease is another one. Yeah.
All right. Any other questions for Abby? All right. Well, thank you so much for giving us a presentation. Thanks to everyone who came, and I hope we will see you in the fall.
Yeah, and thank you guys for having me. This is great.
Thanks so much.